Hi everyone, welcome to this new ESDR Kitchen Freshly Baked session. My name is Sandrine Dubrac, and together with Curdin Conrad, we will moderate this new exciting session. First of all, I would like to shortly introduce the speakers. There will be first Nicole Bertschi, and then followed by Jeremy Di Domizio. This presentation will directly be followed by a Q&A session. And this session is sponsored by Almiral. So we are going to quickly start with the introduction of the first speaker, Nicole Bertschi, who will present her work recently published in Nature Communications and entitled PPAR-γ regulates the effector function of human T helper 9 cells by promoting glycolysis. So Nicole has obtained her PhD at the University of Basel in 2015, where she continued to work until 2018 in a different field than dermatology. And then since April 2019, she moved to the Department of Dermatology of Basel as a postdoctoral fellow. So please, Nicole, you can present your very exciting data. We are looking forward to it. So you are muted.
Yeah. Can you see my screen?
Yes.
Good. So yeah, good afternoon, and thanks for the invitation to present our recent publication about the function of PPAR-γ in TH9 cells here at ESDR Kitchen. So several studies in the past identified a so-called pathogenic subpopulation of Th2 cells causing different type 2 mediated immunopathologies. And interestingly, characterization of these pathogenic Th2 cells revealed common functional properties such as high expression of IL-5 and IL-9, the expression of CRTH2, CCR8, as well as the expression of IL-9 receptor. And in addition, these pathogenic Th2 cells were found to uniformly express the transcription factor PPAR-γ. So in our lab, we recently characterized Th9 cells that can be primed in vitro in Th2 polarizing conditions with the addition of TGF-beta. And to check if these Th9 cells recapitulate the central pathogenic Th2 phenotype, we compared our Th9 transcriptome with 3 previously published pathogenic Th2 specific transcriptomes. And indeed, we found that these Th9 cells share the main features of pathogenic Th2 cells, including the increased expression of PPAR-γ, IL-5, IL-9, and the IL-9 receptor. So we therefore concluded that these Th9 cells represent a good model for pathogenic Th2 cells, and also they represent a good model to study the role of PPAR-γ in human T cells. So to investigate the function of PPAR-γ in these Th9 cells, we chose a transcriptional approach where we incubated the cells in presence or absence of the PPAR-γ inhibitor GW9662. And the analysis of the transcriptome revealed that the PPAR-γ inhibition significantly downregulated all genes involved in the glycolysis pathway, suggesting that PPAR-γ regulates the glycolytic activity in these cells. So interestingly, we found that Th9 cells have a higher glycolytic capacity than Th1 or Th2 cells in the resting as well as in the activated state. And to test whether this is PPAR-γ dependent, we cultured Th9 cells in high or low glucose environments and in presence of the PPAR-γ inhibitor, and indeed found that PPAR-γ inhibition reduced glycolytic activity in the high glucose environments. Moreover, we found that TH9 cells showed a higher glucose uptake compared to TH1 or TH2 cells, which in turn was significantly reduced by PPAR-γ inhibition. And therefore, we concluded that PPAR-γ contributes to the high glycolytic activity of TH9 cells. So to link these findings to effective functions, we next tested whether increased glucose uptake by TH9 cells is also associated with increased cytokine production. And intriguingly, we found that cells with higher glucose uptake also express higher levels of IL-9, suggesting that IL-9 expression is dependent on glucose, while IL-13 levels remained unaffected. Furthermore, we found that IL-9 production could be increased by culturing cells in high glucose environments, whereas again, IL-13 production was unaffected. So to relate these findings to PPAR-γ functions, we again cultured cells in high or low glucose environments, as well as in the presence of the PPAR-γ inhibitor, and found that PPAR-γ antagonism in high glucose environments reduced IL-9 production to the level of low glucose environments, whereas again, leaving IL-13 levels unchanged. So our results suggest that PPAR-γ is a positive regulator of the high glycolytic capacity of TH9 cells, and this high glycolytic activity regulates the expression of IL-9, but not IL-13. So to link this metabolic regulation to the effective functions, we next examined mTORC1. Since mTORC1 was previously shown to be to play a key role in nutrient sensing, and furthermore, the mTORC1 HIF1 alpha axis was previously shown to be required for IL-9 transcription. So therefore, we next assessed mTORC1 activity by examining its direct downstreams target S6, so the phosphorylation of S6. So, and indeed we found that mTORC1 activity was glucose as well as PPAR-γ dependent, and moreover we showed that inhibition of mTORC1 by the known inhibitor rapamycin decreased the production of IL-9, but not IL-13, suggesting again that mTORC1 activity specifically regulates the IL-9 expression. So to bring these findings closer to the clinic, we examined samples from patients with allergic contact dermatitis and found that ACD skin is indeed infiltrated by a high number of phospho-S6 and PPAR-γ double positive T cells compared to normal skin. And in addition, when we analyzed immune cells crawled out from these ACD skin biopsies, we found that PPAR-γ inhibition significantly decreased the activation of mTORC1 as well as the IL-9 expression, reflecting very nicely our results that we obtained in our in vitro assays. So therefore, our results strongly suggest that the glucose and PPAR-γ dependent production of IL-9 in TH9 cells is regulated via mTORC1 in acute allergic skin inflammation. And since it was previously shown that TH9 cells not only express IL-9 at high levels, but also express the IL-9 receptor, we were next interested to study the role of paracrine IL-9 signaling in these cells. And we again chose a transcriptional approach where we incubated IL-9 receptor positive TH9 cells with recombinant IL-9 and performed RNA-seq. And pathway analysis of IL-9 induced genes revealed a coordinated induction of genes involved in the aerobic glycolysis and most prominently SLIC16A1. So SLIC16A1 encodes the monocarboxylate transporter 1, MCT1, which exports the lactate, the end product of aerobic glycolysis. And this transporter therefore plays a key role in the flux control of glycolysis. And at the protein level, we could verify that indeed recombinant IL-9 induces MCT1 expression in these cells. So therefore, we again performed metabolic analysis and confirmed that T cells stimulated with IL-9 show a higher glycolytic capacity, and accordingly, IL-9 increased extracellular lactate levels, which was suppressed by the addition of an MCT1 inhibitor. So together, this data suggests that IL-9 promotes aerobic glycolysis by inducing the lactate transporter MCT1. So considering the crucial role of glycolysis in T cell proliferation, we then next examined the effects of IL-9 on T cell proliferation and found that this is indeed glucose dependent, and this is again abolished by MCT1 inhibition. And we also investigated glucose tissue levels in allergic skin inflammation, and we found that lesional skin of allergic contact dermatitis had higher glucose levels than the corresponding non-lesional control. So together, this data suggests that IL-9 confers a proliferative advantage to T cells in high glucose environments, such as allergic skin inflammation. So in summary, we use TH9 cells that share important features with disease-associated pathogenic TH2 cells to investigate the role of PPAR-γ. And we demonstrate that PPAR-γ controls the high glycolytic capacity, which in turn is a positive regulator of IL-9 expression via mTORC1. Further, we show that the paracrine IL-9 signaling enhances the expression of the lactate transporter MCT1, which provides a proliferative advantage in high glucose environments, such as allergic contact dermatitis. And so together, our data suggests a previously unknown role of PPAR-γ and IL-9 in facilitating immunometabolic recognition of tissue environment. So with this, I would like to thank my supervisor, Professor Christoph Schlappach, as well as my shared co-first author, Oliver Steck and Fabian Luther, as well as the whole team at the dermatology unit at Inselspital and our collaborators. And thank you for your attention.
Thank you a lot, Nico, for this excellent presentation. And to show that Switzerland is representing in ESDR, we have a second speaker again from Switzerland. He actually doesn't need much introduction. He's actually a regular at the kitchen, particularly in the freshly baked. He's publishing too much good results. But nevertheless, I'll try. So Jeremy Di Domizio is now privatdocent, so he has his habilitation recently accepted here in Lausanne, Switzerland, in the lab of Michel Gier. He had his PhD in 2009 from Grenoble, focusing particularly on type 1 interferon and PDCs. First in melanoma, later he was working in autoimmunity. He then joined Michel's, he first joined the Houston, Texas, where he did a postdoc, where he actually got to know Michel and then joined his lab here in 2013, where he's now supervising the laboratory activities of the Department of Dermatology. He's also regular in the ESDR, very active also in the FLA. So thanks, Jeremy, for joining, and thank you everyone for all the spectators, because we have like around 60 people joining despite this unbelievable heat that we're having. And thanks to Almirall again for the support of this kitchen seminar today. So thank you very much for joining, and Jeremy, the stage is yours.
Thank you, Curdin, for this really kind introduction. I would like to thank the ESDR Kitchen committee for asking me to present our last work. So today I will talk about the biology of TH17 cells. So when we talk about TH17 cells and skin, we think about psoriasis, because psoriasis is a chronic inflammatory skin disease characterized by the infiltration of TH17 cells. These cells produce cytokines, including IL-17A, IL-22, and IL-26. And these cytokines can directly activate keratinocytes inducing the hyperproliferation of these cells and the induction of expression of antimicrobial peptides that have pro-inflammatory functions. However, although IL-17A has been extensively studied in TH17 cells, the regulation of expression of IL-26 is unknown. And also, the generation of these TH17 cells require a certain cytokinic environment that includes IL-1 beta, IL-6, IL-23, and TGF-beta. But the role of TGF-beta in this differentiation is not clear. So to further investigate on those two topics, we started to analyze the expression of IL-17A and IL-26 within TH17 cells at the mRNA level by flow cytometry using blood-isolated TH17 cells. And doing so, we observed that there is a subset of cells that express IL-26, but that do not co-express IL-17A, interferon gamma, nor IL-22. So then we wanted to know whether these cells are real TH17 cells. So we performed an in vitro differentiation of TH17 cells from naive T cells using a standard cytokine polarizing cocktail that includes IL-1 beta, IL-6, IL-23, and with or without TGF-beta. And we observed that the differentiation of and the generation of IL-17A producing cells required the full cocktail, including TGF-beta, whereas the generation of IL-26 expressing T cells did not require the TGF-beta. Also, by analyzing this cytokine expression at different time points during the differentiation, we found that IL-26 expressing cells were generated earlier than IL-17A producing cells, indicating that these IL-26 expressing cells may be precursor of the IL-17A producing cells. So to test this hypothesis, we then stimulated blood TH17 cells with TGF-beta. And after a week of stimulation, we could observe the generation of new IL-17A producing cells. But also, there was the appearance of a new subset that co-express IL-26 and IL-17A, demonstrating that IL-26 is an intermediate TH17 subset, and that IL-26 producing cells become IL-17A producers. So this was done in vitro. So then we wanted to know whether this was also occurring in vivo. For this, we switched to the analysis of single-cell RNA sequencing dataset of psoriatic skin. Here we have focused the analysis on dermal CD4 T cells. And we found that IL-17A expressing T cells, here in red, are present in the lesional but not in non-lesional psoriatic skin, whereas IL-26 expressing T cells in green were present in both non-lesional and lesional skin. Also, we found that IL-26 expressing cells were clustered in a different area than IL-17 expressing cells, indicating that they are transcriptionally different. So then to assess whether there is a link between those two subsets, we focused the analysis on the lesional CD4 T cells, and we inferred the pseudotime as well as differentiation trajectory between these cells. And we actually found that the IL-26 and IL-17A expressing T cells are present along the same trajectory of differentiation, that the expression of IL-26 was early and was decreasing over time, whereas the expression of IL-17A was increasing over time, indicating that IL-26 expressing T cells become IL-17A producers over time. And to confirm this, we also analyzed another data set of single-cell RNA sequencing of psoriatic skin that combined TCR sequencing of these cells. And doing so, we could follow specific TCR clonotypes. And we found that in several instances, early IL-26 producers were indeed differentiating into late IL-17A producers. Also, when we mapped the expression of TGF-beta induced genes, we found that IL-17A expressing cells express high levels of this TGF-beta signature compared with IL-26 expressing cells, demonstrating that these cells have been imprinted by TGF-beta, and this demonstrates the requirement of TGF-beta in this differentiation process. So then we ask, what are the cells producing TGF-beta in this skin lesion? So we went back to the full data set, and we observed that TGF-beta was more expressed in lesional compared to non-lesional skin, and that within the lesional skin, it was a subset of keratinocytes that was the main TGF-beta producing cell type. Also, by staining TGF-beta mRNA in psoriatic skin lesion, we could see that those keratinocytes expressing TGF-beta were present in the lower layers of the epidermis, in the area where T cells are infiltrating. Then we wanted to know what does drive this expression of TGF-beta and keratinocytes. So we stimulated normal human keratinocytes with a bunch of cytokines that are usually present in psoriatic lesion. And we found that IL-26 was actually the most potent factor driving the expression of TGF-beta in keratinocytes. This was also confirmed by stimulating ex vivo healthy skin explant with IL-26. So all these data suggest that IL-26 expressing T cells can directly induce TGF-beta in keratinocytes. So to confirm this hypothesis, we then analyzed a spatial transcriptomics dataset of psoriatic skin. As you can see here, we found that the IL-26 expressing spots in this dataset were always associated with high expression of TGF-beta, and they were localized at the dermoepidermal junction. Also, by doing a distance analysis, we found that IL-26 positive T cells were closer than interferon gamma producing T cells to keratinocytes expressing TGF-beta, indicating that there's a link between the T cells expressing IL-26 and the capacity of keratinocytes to produce TGF-beta. And indeed, we confirmed this data at the protein level by performing immunofluorescence staining, where we could find IL-26 expressing CD3 positive T cells in close contact with TGF-beta expressing keratinocytes. So all these data were obtained in psoriatic skin lesion. We then asked whether it was also the case in other inflammatory skin disease, because we and others have already noticed that atopic dermatitis also contain IL-26 expressing T cells. So here we used other transcriptomics dataset that combined atopic and psoriatic skin lesion. First, we found that psoriatic skin lesion as well as atopic skin lesion contain similar levels of IL-26 expressing T cells. However, only psoriatic skin lesion contain a high frequency of IL-17 expressing T cells. So meaning that the differentiation of IL-26 into IL-17A producers only occurs in psoriasis. So why is that? Well, our hypothesis is that the location matters. So in atopic dermatitis, we found that IL-26 expressing T cells were present in the dermal compartment and away from the epidermis, whereas in psoriatic skin lesion, we could see that IL-26 expressing T cells were mainly localized close by or within the epidermis. So this close vicinity between IL-26 positive T cells and keratinocytes can allow IL-26 to induce TGF-beta. And indeed, if we look at the expression of TGF-beta by keratinocytes in all these datasets, we found that only basal keratinocytes from psoriatic lesion, but not basal keratinocytes from atopic dermatitis lesion can produce high levels of TGF-beta. So altogether, we can say that in a non-lesional skin of psoriatic patient, there is the presence of these IL-26 producing TH17 intermediate cells that can go in contact with keratinocytes when they're close by, and when they release IL-26, it can directly stimulate the cells to produce TGF-beta. This TGF-beta will then promote the differentiation of these IL-26 producing T cells into IL-17A producers. And when they release IL-17A, this will lead to the development of a psoriatic lesion. So I would like to thank all the people who participated to this work. This study was driven by Professor Michel Gillier, and Anissa Frias did all the in vitro experiments in this study. And I would like to thank all our collaborators who provided us with the different data sets we have been using. And thank you for your attention.
So thank you very much. Yeah. So thank you very much for these two very nice presentations, which and then we follow by the Q&A. So Curdin, is there any question here?
Yeah, there's one question already coming actually from the panel, very local again from Lausanne. Christine Fisch is asking to Nicole. Maybe the two of you can unmute yourself as well so we can discuss. She congratulates you for a nice presentation, and she asked if you try to activate PPAR-γ with gliatosomes, so rosiglitazones or pioglitazone, for example, and see what's happening.
Yes, we tried to activate PPAR-γ with PPAR-γ agonists, but we never managed to actually push IL-9 levels. And so we then checked for mTORC1 activity, and we actually found that mTORC1 activity was inhibited by these agonists. And literature shows that actually AMPK is activated by these agonists, and which is a negative regulator of mTORC1. So yeah, we think that there's like a parallel pathway that actually inactivates mTORC1, so that we cannot further push IL-9 levels with agonists, PPAR-γ agonists.
I think this is a negative feedback, so it's not getting out of control. Yeah, actually, well, the agonists activate AMPK, and which then negatively regulates mTORC1, so it's like a break within the system then. Yeah, that's what I meant, if it's like a negative feedback loop or an internal break of the system.
Yeah, I have probably, there are some questions. So from Christophe Schlappbach to Jeremy. So he's asking, TGF-beta also controls key elements of the TRM program. Does IL-26 play a role in generation or maintenance of these memory T cells?
So actually, that's a really good question. And what I didn't show is that we also analyzed healthy skin, and you can find IL-26 positive T cells in healthy skin as well. So I wouldn't be surprised that there's a broader role, like you said, to maintain this TRM program in these T cells so that they stay in the skin. But yeah, I don't have more evidence. But definitely the presence of IL-26 expressing T cells in healthy skin says that there's a physiological role of these cells.
Maybe a bit of follow-up on that, if I may. You know, IL-26, where it's usually expressed is MF, and we don't really know what's happening there. And some of them are more TH2, and some of them seem to be more TH17. So you think it could be the same mechanism there, that some of them can induce TGF in the keratinocytes and some don't?
So the thing is, so when we analyzed the atopic dermatitis data set, I tried to see whether the IL-26 expressing T cells could differentiate into another cell type, like it would be like a precursor cells of many cell types, but actually it doesn't happen. So they do not differentiate into IL-13 or IL-4 expressing T cells. So in my analysis, I only see the TH17 pathway. But then, yeah, so for MF, it's more difficult to answer. Maybe the IL-26 expressing T cells that is resident become the tumor cell in MF. And then there's a bystander activation of Th2 cells. Maybe that's why people think that there is a Th2 polarization in MF. This is not clear to me.
I was asking because it seems a subpopulation that actually drives the TH17 response. And in rare cases, so that these might be ones that have this reaction that you just showed, that they can actually, their keratinocytes can induce TGF-beta, and this then induces this TH17 response in these cases.
Yeah, so the absence of TGF-beta triggering, would keep them in another state. That could also be an explanation.
To make the Q&A happen, I have a very short question to Nicole. When you observe increased uptake of glucose in your cells, did you measure expression of GLUT isoforms, so the molecules which are uptaking glucose from the medium, from the outside?
Yes, we checked it in the RNA-seq, but actually there's not a increase in these GLUT transporters.
Shall we move on to...
Maybe very, very quick question from Yongchu to finalize the questions that we have in the chat. Again, to Jeremy, do you think that IL-26 might be higher in Asian AD patients? That might explain their different cytokine profiles.
Yeah, so this I didn't check specifically, and I have to say that when we analyze data sets from a patient, we don't have always the ethnicity of the patient that were used. So it's hard for me to say. I'm pretty sure there are data sets somewhere that compare. So, but I don't have the answer.
Okay, thank you very much. So we can try to move on to the end of this session and to introduce the next presentation, which will be the bench to bedside skin session, and it will be a talk on the skin barrier diseases by Matthias Schmuth from the Medical University of Innsbruck, and we are really much looking forward to his presentation. And I would like also to emphasize the next ESDR meeting that will take place in Lisbon, Portugal, from the 4th to the 7th of September 2024. And we are waiting for you to attend this meeting, very numerous participants. And this being said, I would like to thank you all for your attention, and Curdin for co-managing this session.
Thank you. Let's have an ice cream. And maybe just very briefly, thank again Almiral for the support, and wish you all a happy end of the summer. Thanks a lot. Bye-bye, everyone.
Ciao, everyone.